Chapter 5 is titled The Working Cell. We learned in the last unit that phospholipids make up the plasma membrane, also known as the cell membrane. So just a reminder of the structure, there is a glycerol head, and attached to the glycerol is a phosphate group and another molecular structure attached to it. We don't need to know the name of it. There are two fatty acid tails. Looking at the structure of this head, it is hydrophilic because there are these electronegative oxygens that are going to hog the electrons, create partial charges, and give a polarity, and polar is a synonym for hydrophilic. The tails are hydrophobic because if we look at them, they're made of just carbon and hydrogen. These are non-polar bonds, so they're hydrophobic. We are going to symbolize a lot of times these phospholipids with this tiny little symbol where you just see this is representing the head and then the two fatty acid tails. Here's the way the phospholipids come together to actually form the plasma membrane. We say that it is a phospholipid bilayer, where bi means two, because you can see we have two layers of these phospholipids. The way they come together, the hydrophobic tails of both layers, they're going to be in the middle where they can kind of crowd together and exclude water because they're hydrophobic. The heads are on the outside, where if this is the head, and let's say that we have the outside of the cell out here, well, for the most part, cells are in water-based or aqueous solutions. So these hydrophilic heads, they're going to be good facing the outside of the cell. And then if the other side is also facing a water-based solution, which would be my cytoplasm of the cell, again, those hydrophilic heads are okay with being near the water of the cytoplasm. Because of the hydrophobic interior of the plasma membrane, we can say that the plasma membrane is selectively permeable. That means that some molecules can pass through more easily than others. If I just look at the width of the plasma membrane, a majority of it is these hydrophobic tails. So hydrophobic or nonpolar molecules, they can pass through more easily because it's mostly those tails that they would have to navigate through to get all the way to the other side. Now the opposite, polar molecules or charged, so ions, they are attracted to water. They're not lipid soluble. They are not going to easily be able to cross the phospholipid bilayer. Size is also a factor, so smaller molecules can pass through more easily than larger molecules. The plasma membrane is known as a fluid mosaic model. So first I'm going to talk about why is it called a mosaic. Looking at the plasma membrane, it is not just phospholipids. There's a mosaic of other molecules in there. So for example, there are proteins embedded in the plasma membrane. Some of the proteins have carbohydrates attached to them, which now I can call those a glycoprotein. Glyco means sugar. I have cholesterol in my plasma membrane. I can have lipids attached to even the outside of my phospholipids. Inside the cell too, there are these structural supports called the cytoskeleton. Outside of my cell, there is more structural supports that are part of what we call extracellular matrix. Extra means outside of. So we can see there's a lot going on here. The reason why we call it a fluid mosaic is that these molecules are not static. They can move around. So just because this protein is right here doesn't mean that it can move to other parts of the plasma membrane. Let's take a look at the function of cholesterol in the plasma membrane. We did learn that cholesterol is a type of steroid, and steroids are a type of lipid. What makes something a steroid is that it is a lipid with a ring structure to it. The purpose of cholesterol in plasma membranes is it helps stabilize the phospholipids. So yes, there's fluidity with the phospholipid bilayer, but then I do have these cholesterol molecules kind of wedged in there to give the plasma membrane some stability. And also it makes sense when you look at where they are. Cholesterol is a lipid, so it likes to be near the hydrophobic tails of the phospholipids. There's a variety of functions that proteins can have that are wedged in the plasma membrane. So this is telling you that proteins can act by attaching the cytoskeleton in the inside of the cell to this extracellular matrix outside the cell, and it really gives cells stability. Proteins can function in signal transduction, where basically if I have a signaling molecule like a hormone, it will attach to this receptor protein and then send a signal inside the cell. Some proteins work as transport to help move things in and out of the cell. Intercellular junctions, inter means between, so there are some proteins that are going to attach one cell to another. This is especially important in animal cells that don't have cell walls to bind their cells together. There's also proteins working with cell-to-cell recognition so that one cell in the body can recognize the other cell and know what its specific function is, meaning like a liver cell versus a heart cell. The cell-to-cell recognition, you can see that a lot of times it is a glycoprotein, meaning a protein attached to a little carbohydrate or sugar molecule. And then you can also have proteins function as enzymes in the cell membrane. Let's take a look at how molecules move in and out of cells. First, the word diffusion is just referring to the random movement of molecules. Molecules always want to be spread out evenly in order to reach equilibrium. So the way they're going to do that is they're going to move from an area of high concentration to an area of low concentration. Passive transport refers to diffusion across a membrane. And now if I'm just talking about diffusion, the way molecules naturally move, they're just moving from high concentration to low concentration. And because this is the natural way molecules move, this process does not require energy. So if I take a look at this picture, I can start with seeing a membrane. And then there is a concentration gradient of these molecules of dye, meaning there is a higher concentration on one side and a lower on the other. This is called a concentration gradient, a difference in concentration. So my molecules of dye are going to move from an area of high concentration to low concentration. We can see that happening until they reach equilibrium, where there is an equal concentration of molecules on both sides of the membrane. I want you to see that in equilibrium, since molecules are always moving, that even when we have equilibrium, molecules will still move, but now they'll move at an equal rate back and forth on either side of the membrane. This picture is illustrating that passive transport does occur independently with different molecules. So if the green is oxygen and the purple is carbon dioxide, oxygen will move this way down its concentration gradient from high to low concentration. Carbon dioxide will move the other way until, again, they reach equilibrium, and then both molecules will be moving at equal pace back and forth through the membrane. We already mentioned that some molecules cannot move directly through the phospholipid bilayer. Those things, ions and polar molecules, they need to have a facilitator. They need to move through a membrane protein that is in the phospholipid bilayer. So this process is called facilitated diffusion. It is still a type of passive transport because it does not require energy, since molecules are still moving from an area of high concentration to an area of low concentration. These transport proteins, however, are specific to the molecule it can carry. If this is a transport protein for sodium ions, for example, it can only allow sodium ions to pass. Osmosis refers to the facilitated diffusion of water. Now, water is a polar molecule, so polar molecules do need membrane proteins to move. The membrane proteins that water crosses, they're called aquaporins. Water can sneak through membranes. It's just that since they're polar, it does take longer for them to move directly across the membrane. Now, the way that water moves, it is determined by total solute concentration. Solute is anything that's dissolved in water. What I mean by total solute is that water doesn't look at, let's say, salt separately from glucose, all right? It's just going to be determined by total solute concentration. I can see here that water always moves to dilute the higher solute concentration. So in this tube right here, if I have a selectively permeable membrane where water can sneak through but the solute cannot, water is going to move from an area of low concentration of solute to high concentration of solute. It's going to try and dilute all of these solute molecules, and then it's going to do that until I have equal concentration of solute on both sides of the membrane. You can also think that water is moving from an area of high water concentration to low water concentration. Concentration of solute versus water is always going to be the opposite. When I think about cells and where water is going to move, I have to compare the concentration of the cell and the outside of the cell. If a cell is in an isotonic solution, that means that the solutions have an equal solute concentration. So if this is a cell, this is a red blood cell, if it is in a solution that is equal concentration to inside, I am still going to have movement of water because molecules are always moving, but there's going to be an equal concentration of water moving in and water moving out. So in this case, my cells are not going to change in size. A hypotonic solution refers to a solution with a lower solute concentration. So the way I remember hypo, I hear hypo and then I think low. So if my red blood cell is put in a solution that has a lower solute concentration, then that would mean that inside the cell there is a higher solute concentration. Remember, water always moves to dilute, so water is going to move from the lower solute to the higher solute. The issue with this is that if too much water enters, my cells can burst, and that is called cell lysis, or I could say my cell has lysed. A hypertonic solution refers to a solution with a higher solute concentration, so the prefix hyper means higher. If I put a cell in a hypertonic solution, that means outside the cell there is a higher solute concentration, which means inside the cell is a lower solute concentration. And again, since water always moves to dilute, now water is going to move from lower to higher solute concentration. It's going to move outside the cell. So now my cell is going to lose water and it is going to shrivel. Now let's compare what happens to animal cells and plant cells in these three different types of solutions. Everything I've showed you so far is what happens in an animal cell. Where animal cells, they really are healthiest in an isotonic solution. In this case, water is moving in and out of the cells at an equal rate because the cells are in a solution that has equal concentration to them. Animal cells, we don't want them in a hypotonic solution because if they gain water, they can burst. We don't want them in a hypertonic solution because they will lose water and shrivel. It's a different case with plant cells. Plant cells are actually healthiest in a hypotonic solution. Plant cells, they want this influx of water. they will not burst because they have the cell wall that makes them rigid. So when they take in water, they swell. Some of that water can be stored in their central vacuole, and that's actually what helps keeps plants upright. In an isotonic solution, water is still moving in and out of plants' cells equally, but because there's not that net influx of water, there's not that pressure on the cell wall, so we can say that these cells are flaccid, and that's what's going to make the plants wilt. It's really the same thing in a hypertonic solution for plants, is that they also don't want a net outflux of water because the cells will shrivel, and something will happen called plasmolysis, where basically the plasma membrane starts to pull away from the cell wall. Aside from passive transport, there is active transport. This is when we are moving substances against the concentration gradient. So we're moving things from low to high concentration. We're basically trying to stockpile molecules on one side of the membrane. Since this is against the natural way molecules move, it requires energy. Active transport also does require a membrane protein. Just because the energy molecule that cells use, ATP, it needs something to bind to. Bulk transport is if we want to move a lot of molecules at a time, and if those molecules are large things such as proteins. Exocytosis is specifically when we want to move molecules out of the cell. We can think molecules are exiting exocytosis. So those molecules will start by being enclosed in a membrane-bound vesicle. That vesicle will move closer to the plasma membrane. The vesicle will start to fuse with the plasma membrane until the molecules are actually released. Endocytosis is when we want to move a bunch of molecules or large molecules into the cell, so endocytosis enter. It is the opposite process of exocytosis. Even in this picture, you can see that the membrane starts to fold in, the molecules kind of collect in there, and then this gets thinner and thinner until it pinches off, and now we have a vesicle inside of the cell, and we've brought those molecules in. There are 3 types of endocytosis depending on what is being brought into the cell. Phagocytosis is when either food or other type of solid particles is being brought into the cell, such as a single-celled organism bringing in its food particles. Penocytosis is when liquid is being brought into the cell. When things are being brought into the cell, if it's a larger thing, we can call it a vacuole. If it's smaller, then it would just be a small membrane-bound vesicle. Receptor-mediated endocytosis, this is when a specific molecule has to attach to a receptor before endocytosis occurs. So for example, cholesterol, we do want to bring that into our cells. We don't want it to be in our blood. We want to bring it into our cells so that we can incorporate it into the plasma membrane. So what would happen is cholesterol would bind to a receptor, and then that would initiate endocytosis, and now these cholesterol molecules will be in vesicles inside of our cells.